So Andy, maybe you could introduce your great friend, Britt Bunyard.
I should be delighted to, Mr. President. Thank you for the invitation. Okay, well, let's go to a slideshow here, shall we?
Sure.
There were some questions from members of SVIMS about whether Britt was handsome also when he was younger. And here is a picture of the good Dr. Bunyard as a graduate student at Penn State University. The year is 1995, and the creature crouched behind him. I believe, correct me if I'm wrong, Dr. Bunyard, this is the famed Nittany lion, is it?
Oh, yes.
The mascot of Penn State University.
Oh, yeah.
A very, very fancy name for a cougar or puma. The last one of which was killed in Pennsylvania in 1856, I believe. But anyway, a graduate in 1995 of the Nittany Lions, the Penn State University. Dr. Bunyard is founder, publisher, editor-in-chief of the Mycology, the International Mycology Journal Fungi. If you don't have a subscription to it, maybe I'll just pause right now so everybody can subscribe while I'm doing these. But you ought to have a subscription to it. It's a spectacular monthly or quarterly treat. I love receiving it in the mail. And very reasonable and featuring all kinds of wonderful articles. He's worked academically as a mycologist his entire career, teaching at a number of university courses, writing scientifically for research journals, popular science magazines. He served as an editor for both mycological and entomological research journals and mushroom guidebooks. I like to work with mushrooms and plants. I know crossing kingdoms brings huge riches, and I know Britt loves the interactions between insects and fungi. He's certainly a popular evangelizer on all things fungal. He's been featured on National Public Radio, All Things Considered, PBS's Nova, and the Wisconsin Foodie television programs, and has been interviewed or quoted in Discover Magazine, The Atlantic, Vox, Vogue, Forbes, and a whole host of other esteemed journals. He's the executive director of the Telluride Mushroom Festival. Here he is at a mushroom tossing competition, I think in Colorado, not sure. And this has got to be in Telluride. I can't imagine it's anywhere else. He's authored a number of excellent books, including Amanitas of North America, The Beginner's Guide to Mushrooms and Macrofungi of Ohio and the Midwestern states, and wait for it and save up for it, the forthcoming The Secret Life of Fungi with Princeton University Press. When's that coming out, Brett?
In about 30 days.
About 30 days.
Yeah, you can pre-order now. I guess it starts shipping maybe in 30 days.
Fantastic. It's going to be a brilliant work. I'm certain of it if it's anything like his previous publications. And then in 2021, Britt was awarded the Gary Linkoff Award for contributions to amateur mycology by the North American Mycological Association. It's NAMA's most prestigious honor for American mycologists, and it's really an honor to win that prestigious award. But Britt is also a good friend to British Columbians and a friend of the South Vancouver Island Mycological Society and the Machosen Biodiversity Project. Here he is at the Machosen Myco Blitz in 2014. Kim Luther in the center, Britt and myself in the back left, Adolf and Aluna, Paul Kruger, and lots of other people you'll know in that picture, I think. James and Juliet, lots of good SVIMS members. Here we are in the Machosen Council chambers trying to identify some mysterious mushrooms. In the field, Daryl Thompson, me and Britt trying to sort out some LBM or other. I can't even remember what it was that we were working on at the time. Here he is looking at Agaricus augustus in the Machosen Myco Blitz. Britt is no stranger to SVIMS, no stranger to British Columbia, and an eager and enthusiastic and terribly fun participant in our forays and bio blitzes and Myco blitzes and the like. Here he is at Blinkhorn Park on a SVIMS foray, holding the swamp lantern scepter of truth while preaching to the assembled multitudes. So please join me in welcoming Britt Bunyard to our SVIMS meeting.
Great, thank you, Andy. Thank you very much. And Andy, I don't know if you heard what Britt had to say. This is going to be his first time doing a Zoom presentation. If I had known, I would have bugged him a bit more and done some practice.
Yeah,
but he's
no,
never home.
It's not actually the first time. It's just the first since about the first couple months of the pandemic, 2 or 3 years ago, or however long it's been going on. I did several right away, and then I just decided to stop doing them because I wasn't enjoying them so much, and there's been some, everybody was having problems early on, kind of figuring it out. So I just kind of swore them off, and also I was kind of figuring the pandemic was going to end, and so I also thought I would brew beer every 2 weeks for the pandemic. A whole bunch of home brewers across North America signed on to that. So I'm now more than 40 batches of beer later and a lot of pounds heavier. I'm still brewing, but eventually it's going to end. So, well, yeah, thanks for inviting me to do this. We've obviously been trying to hook up for a while. Also, thanks to the one astute viewer of Andy's photos there that noticed there was a Bernie Sanders in the audience in one of those photos. Maybe only one person noticed. That was during the whole Bernie Sanders thing. So I've been, back to the Pacific Northwest, even during the pandemic, and trying to hook up with SVIMS for a while now, but we keep running into problems. And so, so I just recently hopped in my truck there and was going to drive out to do the presentation for you guys. So I live near Chicago, so I thought, well, I'll head due north and hop on the Trans-Canada and maybe even stop off in Ottawa for a while, see that wonderful capital city. And I don't know what was going on recently, but there's a lot of people in Ottawa. And so I parked my truck for just 2 minutes, or maybe it was 2 weeks, I don't know, but it got towed. Everybody's trucks were getting towed. There were trucks everywhere. I don't know what was going on. So I decided to give up, came back home and chatted with David, and we said, well, let's just do this Zoom style. And so hopefully this is going to work. But anyways, that was my latest attempt to try to make it out to Vancouver Island. David said he wanted to hear about something other than Amanitas or the usual. And when he got wind that I had this book coming out soon, he said, let's hear about that. So here's a cover of the book coming out by Princeton University Press. It's you can see about it on Amazon as well as at their website right now. Also, you can pre-order it now. If you pre-order through the website, you see there's a big fat discount of 30%. I asked them if it works for Canada, and they said, yeah, it works the same. They have I guess a distributor up there. So you can save some money off of the book. Anyway, so it'll be tales from this book that we'll be hearing about tonight. So this book about fungi is in some ways similar to most other books on fungi out there, in that there's a lot of basics about fungi, physiology and what they are and how they reproduce and all that sort of stuff. But there's a big focus on a lot of fungi and a lot of oddball stories that most people have probably never heard, and for sure some stories that nobody's ever heard. So no matter how astute you are with mycology, there's probably something in there for you. In any case, it starts out with fungal basics, what are fungi? And it seems like a pretty fundamental question that everyone would know, but actually I get asked all the time, and everyone knows, or at least thinks they know what a fungus looks like and thinks they know what a fungus is if you were holding it in your hand. But, there's a lot of fungi that don't make mushrooms, so how would you really know? So if you don't already know, this is a little quickie overview of what fungi are. They're eukaryotes, first off. So, okay, well, what's a eukaryote? Well, all living organisms on the planet are either prokaryotes or eukaryotes. Bacteria are prokaryotes, which are single-celled organisms that don't have organelles, basically, in their cells. That's pretty much the overview. So they don't have chromosomes, they don't have mitochondria, anything like that. A bacteria basically functions as a mitochondria, they're quite small. So all the other life that's not bacterial are eukaryotes, bacteria prokaryotes. So the eukaryotes are things like fungi, animals, plants, and protists. So within the group of eukaryotes, fungi and animals are most closely related. They come from a common ancestor. So a lot about fungi are quite similar to animals. A number of things about their cellular physiology, chemistry, etc., enzymes, is very, very similar to animals. A lot's quite different too, but a lot's quite similar. So some other things about fungi is they're extremely diverse. Again, the vast majority of fungi do not produce structures like mushrooms, and you really wouldn't even recognize them if you saw them. Most are quite small. Unlike most organisms on the planet, fungi exist in every biome on the planet, from marine environments, freshwater, terrestrial, even the driest places on the planet have fungi, even places where a lot of other life doesn't exist. And then the other thing about fungi is it's the study of them is kind of a new thing. So there's still a lot we don't know about them. They're poorly known, and most species of fungi are probably not even described yet. As an example of just how diverse they are, you can find fungi in the driest places on the planet, like Antarctica and deserts and places like this. These scenes here from Antarctica, and it's too cold and dry and inhospitable for almost any type of life whatsoever. You see in some of those pictures maybe a few types of plants, there might be a few types of arthropods, and that's about it. There's not a lot of other things that can exist there. It's just too harsh. But you'll find fungi there, despite how dry and cold it is most of the time. They aren't necessarily going to be out in plain view. Some of these fungi are lichenized type fungi, which is a symbiotic situation with a photosynthetic organism. And these lichens actually oftentimes aren't even on the surface of rocks because it's such harsh conditions. They actually exist down inside the rock, just under the surface, believe it or not. You see where those red arrows are, they're pointing to some of these lichens that can persist in these porous rocks, which is kind of cool. So again, in places you can't imagine a lot of things to live, certainly things that seem to us to need to have very moist inhabitants, moist habitats, they can find a way to make do there as well. So pretty neat. So besides a lot of the wacky tales and things like that, the two main themes of this book are shown here. Basically, they are to point out that much of what we thought we knew about fungi, once people start looking into them, find that it ain't necessarily so. There's a lot of different fungi that have been known for a long, long time. It was well understood what they did and how they acted, or at least we supposed we knew what they did and how they acted and what their ecology was. But upon further investigation, even very recently, a lot of very common fungi have been found to have things going on that we didn't know. I didn't sit in on the hygrophorus talk that was just mentioned earlier by David, but undoubtedly it was mentioned that some of the hygrossa bees, super common organisms, you see them on lawns all the time. It was only recently discovered, just a few years ago, that a number of them are mycorrhizal with grasses and things like this. We thought we knew what they were doing in the habitat. We thought that they were saprobes. Turns out they're not saprobes. They live sort of an endophyte inside the grass and come out through the roots and have this kind of a weird sort of mycorrhizal association, then produce a fruit body. So hidden right in plain sight. And that's sort of the other theme of the book as well. There's a number of fungi in the book that are focused on that everyone would recognize, death caps and things like that. And you have a fungus book, you have to have these mushrooms that everyone recognizes, but probably 80% of the stories in the book are fungi you've never heard of. A lot of them are small but have amazing things about them, or they've changed the course of the world, or continuing to change the course of the world now. So these are sort of the two main themes of this book. So although at first glance you might think, well, do I have room on the shelf for yet another fungal book? But hopefully I think I've put together something that's very different from all the other books on fungi out there. The subtitle of the book, A Natural History of Our Planet's Decomposers, is unfortunately a really poor title, and I didn't have a choice there. The title was chosen before they asked me to do the book, because if you pick up a copy, you'll soon see that the vast majority of the fungi in the book actually are not decomposers, and that's just because that's the way fungi are. More on that in just a couple seconds. So one of the sections having to do with fungi, which is quite interesting, has to do with their chemistry and physiology, because fungi can do really amazing things. They've been put to use for industrial purposes, and people brew with them and do all sorts of things. But fungi in nature, they're doing what they do because that's their lifestyle. And to me, it's just fascinating, some of the things that fungi can live on. So again, going back to fungi being closest relatives to animals, they have a similar lifestyle to animals. Unlike plants and a number of bacteria that are autotrophs, all fungi are, so far as is known, are heterotrophs, just like all animals, which is to say they can't manufacture their own food by photosynthesis or chemosynthesis or something like this. They have to rely on consuming other organisms that were living or already dead for their energy and carbohydrate source, etc. So a lot of fungi are saprobes. We learned this in school, of course. Everyone knows, fungi rot things, and it's really fortunate they do. Otherwise, all this plant matter would pile up on the earth and we'd be suffocated under it. And well, turns out that's not really true. Of course, some fungi are saprobes, especially the ones that we can cultivate, but the vast majority of fungi are not saprobes. They do other things. They're mostly biotrophs. So, but the ones that do have this saprobic lifestyle, you've seen these before. They're pretty well known to everyone. So I'll just show a slide or two here. But I want to point out, when you see rotting wood in the forest, you'll see it has sort of one of two looks: this kind of brown blocky material or this kind of white stringy material. And most people, when they see that, they're like, oh, okay, I see this brown blocky material. And I see a lot of that up in the Pacific Northwest. Well, that's because when conifers rot, that's how they rot. They fall apart in these neat blocks, and some hardwoods do the kind of white stringy thing. And actually, it doesn't really have so much to do with the type of tree it was. It actually has pretty much everything to do with the fungus that's attacking the wood, believe it or not. So what makes up plants, when you see a tree, almost everything that you're looking at in this big trunk of a tree, maybe a big Douglas fir, almost everything you see there, believe it or not, is dead. The outer skin of the tree, that's living, and the leaves and things, that's living. But all that heartwood that's many, many years old, that's dead wood that was once living and now it's dead. All that material, the other astounding thing is all that material, none of it came from the soil. All of it came from the air. It was all fixed by some enzymes in the plant leaves and needles when they were photosynthesizing, and that's where all this carbohydrate material came from. That's kind of astounding to think, considering these trees can weigh, hundreds of tons. But in any case, what makes up a plant is mostly two main compounds, lignin and cellulose. So the cells of plants, The cell walls are primarily made of cellulose, and what holds the cells together is sort of this cementy material, lignin. So it turns out to break this stuff down takes a lot of energy or some other sort of mechanism. A lot of fungi have figured out they can have enzymes, they can develop enzymes that can break this stuff down and make use of it. But both of these compounds are so involved that really fungi can break down one or the other. They can't really break down both all that well. So the fungi that break down wood and leave behind this kind of brown blocky material, the lignin, those are known as brown rot fungi. A number of them are mushrooms that you and I would recognize, Fomitopsis pianicola and Phalinus, etc. If they break down the lignin, as well as the cellulose somewhat, but leave a lot of the cellulose behind, you'll end up having this kind of white stringy looking material left over. Those are the white rot fungi. So things like armillarias and ganodermas and trimides, they're white rot fungi. So they're really good at breaking down the lignin, sort of good at breaking down the cellulose, but not real good. And in fact, a lot of people think they're probably mostly breaking down the cellulose just to get it out of the way so they can get at the lignin, because they can apparently get a lot of energy from that. So in any case, these are saprobic fungi, and these are what they do. They do some pretty cool things besides just merely breaking down wood and turning that back into soil, etc. They do some things along the way involving enzymes and some neat chemistry, and frankly, it's almost like magic what they do along the way. If you saw the tree before it fell down in the forest and started rotting, it looked like pretty much any other living tree. So here's a species of populus with some Phalinus polypores growing off the side. This Phalinus, it's pretty well camouflaged. A lot of people would walk right by this and not even notice those were fungal fruit bodies. In any case, the Phalinus got inside the tree, probably through some sort of a wound in the side of the tree, a lightning strike or a branch broke off, something like that. Fungus gets inside the tree and starts rotting the heartwood of the tree, that dead wood that's on the inside. The tree probably doesn't mind all that much because the wood's dead, and as long as the fungus stays put, the tree's not really in much danger, and it'll live out probably its entire life without really succumbing to any sort of a fungal invasion. And the tree actually even does stuff to keep the fungus contained. The fungus also does stuff to keep its area contained, and you'll see it by these black lines that get in wood. If you were to rip one of those conchs off the side of the tree and look what's going on the back side where it's attached to the tree, this is what you would see in the picture on the left. You'd see these black lines going down into the wood. You see that black, almost kind of corky looking wood. It's kind of brown black on my screen. That's all kind of partially rotted heartwood there where that conch's sticking off the tree, and where the fungus actually starts invading the wood there. Again, it's not necessarily living wood. It's going to be this heartwood. The fungus sort of wants to wall off its area and contain it and keep other fungi and other microbes that are also eating away at the wood, keep them out. So it lays down this sort of layer of melanin. And we have melanin, of course, in our cells. Melanin is a compound that lots of organisms use to fend off all sorts of attack from outside, chemical onslaught, UV light, severe oxidation from any number of things. Melanin is used by a lot of organisms for a lot of purposes, basically for protecting the cells. And so fungi can melanize this area of wood, and the fungus is now inside this, if you can imagine this sort of 3D chamber that's inside this melanin. And so now the fungus can go ahead and continue living out the rest of its days in there without being bothered so much by other things getting at it. What's really cool is people harvest this wood and turn it into all sorts of objects, like this bowl here, which is quite pretty and even has a number of different fungi attacking it, such that different fungi can impart sort of pink and gold and green areas besides the black lines in the wood. And then this pipe here that a friend of mine made for me that has all this beautiful patterns from the wood rot fungi. You'll hear wood that looks like this referred to as like bird's eye maple or curly maple, or there's all sorts of names. People make cabinetry, guitars, furniture out of bird's eye maple and curly maple and green oak and stuff. There's no such thing as bird's eye maple or curly maple. It's all sorts of different types of trees that have this fungal this fungal attack going on, and results in these pretty patterns. But here's the neat thing: while everyone's seen the wood that has this stuff happen to it, lots of other plant material has the same thing. You can see in this Lophidermium here that gets on pine needles, it also makes these little black lines of melanin to wall off its little zones. Those black pustules are the fruit bodies of this tiny little fungus that lives in pine needles, and separating each one of those colonies are these black zones of melanin laid down by the fungus. Isn't that pretty cool? Most people would have probably never seen that, but if you're down along the coast and there's a lot of tall kind of reed looking plants that get mycenas and things rotting on them, take a look at this dead material, and there'll be all kinds of different fungi growing in this dead debris, and a lot of times they'll leave these black lines, these striations in the material. So pretty neat to see. Okay, change gears to reproduction. Pretty fascinating, the reproductive modes that fungi go into. And again, like animals, fungi reproduce primarily by sexual reproduction. Like animals, not all of them reproduce sexually. There are asexually reproducing animals, and there are asexually reproducing fungi. In fact, probably most fungi most of the time reproduce asexually, and a lot of fungi don't seem to reproduce sexually ever. At least we don't see any signs of it, like mating type genes, etc. So it's not entirely known what some fungi do. Frankly, I don't know why fungi go to the trouble of reproducing sexually at all. They can do a number of other things to bring about genetic variability, etc. But besides sexual reproduction and asexual reproduction, a lot of fungi just sort of grow and have vegetative propagation. There's fragmentation, and particles of the fungi move around with soil and things like this, is how a lot of them get around. A lot of the very common molds in your house that rot things, those are mostly asexual fungi, and they don't produce sexual spores. They produce what seems like a spore. It's a little propagule, but it's really nothing more than clone from the parent. So the fungus grows as a mold throughout fruit in your refrigerator or cheese or something like that. And then once the colony is mature, it just basically rears up in fragments, or if it's really elaborate, the fragmentation looks like little propagules and then they blow away. Aspergillus and Fusarium and just all sorts of very, very common, widespread saprobic and pathogenic fungi reproduce this way. As far as mushroom-producing fungi, which is sort of a minority of all the fungi, number of them can reproduce asexually. It's not really been looked at too well. It wouldn't surprise me if all of them can reproduce asexually as well. And, to be completely honest, sexual spores for mushrooms, very few of those probably ever amount to anything whatsoever. The vast majority are made, and they're probably pretty cheap for the fungus to make, and they go to waste, and it's not really a big deal. So who knows what percentage of the time the fungi are getting around by asexual reproduction, but with a lot of them, it seems to be the primary way. So, and along those lines, the thing about asexual reproduction, back when I learned fungal taxonomy, of course, there were, four main phyla, the chytrids and the zygomycetes and the ascomycetes and the basidiomycetes. And then there was this whole big other group, the fungi imperfecti, the fungi that reproduced asexually. We didn't really know how to classify them because classification of fungi is based on the sexual reproductive structures, just like with all organisms. If you know about plants, they're grouped by what sort of a flower they make. All leguminose things produce the same type of a flower, rosaceous things, et cetera, et cetera. And so fungi are classified the same way. Well, now with DNA sequence technology, of course, scientists can figure out that this fungus that only seems to reproduce asexually, it's a very important plant pathogen, economically important, we need to know everything about it. Well, they've been able to determine, well, this fusarium actually turns out to be an aspergillus or something when it does, this fusarium or this aspergillus, when it does its sexual reproduction, turns out it's an ascomycete, and this is, what we would call it by its teleomorph name. But in any case, another thing that's crept up into the picture with the use of DNA sequence technology, we've determined that a lot of stuff that was zygomycetes and even some of these other groups really didn't belong in that group. And there's also these other fungi we've been finding in the environment that we can't seem to culture them. They don't seem to make fruit bodies. In fact, when investigated, they don't seem to reproduce sexually at all. They've all been placed in this group called the Glomeromycota. They're, I don't know how many different species we're up to now. It's not very many. I think it's just a couple hundred still. When I was a student, there was only, I think, a few dozen known. They're mostly known from DNA sequences, and most don't seem to reproduce sexually at all. So they don't produce spores or fruit body or anything. In fact, it's not really known much about what they do, except that they're mycorrhizal with pretty much all plants on the planet, and everywhere we look, we find them. So the interesting thing about them is we know almost nothing about them, but they're probably the most important fungi on the planet. In fact, they might even be the most important organisms on the planet because they are mycorrhizal with just about all the terrestrial plants there are. So although we don't know a whole lot about them, it seems like they're the puppet masters of maybe the entire planet. So I suppose if you come back in another century, we'll be having forays to look for them, and everyone will be really excited and asking the same questions like, can you eat it, et cetera, et cetera. But it'll be with these really tiny things in the soil that don't produce, really big, beautiful, elaborate fruit bodies. But since most of us are more familiar with the big, elaborate fruit bodies, we can turn now and look at them. The vast majority of fungi are not basidiomycetes. It kind of seems like they are because those are the ones that we bring into forays and everyone's excited about, but they're actually kind of a small group. The vast majority of fungi are actually ascomycetes, and they're, plant pathogens and endophytes and things like this. But anyways, the basidiomycetes, everyone likes to see them because they are rather big and showy and pretty. And they also have a really cool form of reproduction. So I sort of failed to mention, the way fungi are classified is on their sexual reproduction. Well, the chytrids, they produce kind of this little, the ones on the far left, they're the most primitive fungi. They produce sort of an oogonium, and they have a swimming zoospore. The male part swims to it, and there's fertilization. And that's, not super exciting, and it's quite small. You certainly don't go looking for them on forays. The zygomycetes, arguably, they're even less exciting. They produce sort of this zygospore, and when two different mating type molds kind of get together, and that's pretty much all they do. The ascomycetes, they do all kinds of things, a lot of little teeny ones. They can make some really pretty structures. Some make large structures like morels. So those are kind of cool, but their sort of squirt gun method of shooting spores into the environment, well, kind of cool. It really can't rival the basidiomycetes. So although they're not the largest group, they certainly have some really amazing things they do when it comes time to reproduce. So basidiomycetes include things like polypores like these guys, and then also gilled mushrooms like these shiitakes. But the same thing's pretty much going on no matter if they have tubes and pores or gills. What's going on, if you take a look, is on the surface of these gills or the walls of the tubes of polypores and boletes is there are Bastidia, the club that they get their name from, arises from the tissue of the gill, and that produces the spores. And, the average person assumes that spores drop from the caps of mushrooms and waft away. And you could even see videos of this online. But there's really more to it than just that. It's not a passive thing like the mushrooms are dropping. There's actually tremendous force to blast these spores away from the surface of the gills or the tubes. And there's even some other things that go on besides that have to happen before this is going to work. If you pick gilled mushrooms or boletes or polypores and put them in your basket upside down, they can't release the spores. That's just the way it goes. They have to be upright, and the cap has to be horizontal with the surface of the earth. So you see in this cartoon here why those shiitakes grow the way they do. If that one on the bottom there coming out exactly perpendicular, and it was to be blasting spores, the spores would all blast and then fall and land and stick to the gill on the opposite side, and none of them would be successful. So the cap has to come out and be kept horizontal with the surface of the earth for the spores to be blasted from the surface and then fall down between the gills, and then the wafting and the germination and all that happens. But there's even some other cool things that happen besides that. If you've collected a lot of different mushrooms, you've probably seen some things like this. The bottom left just simply shows a type of an oyster mushroom coming out I think that's a Panislevis, coming out the side of a tree. And, sure enough, it has to curve its stem upward to keep the cap horizontal with earth. If you've collected Amanitas, you've probably seen that unless you store them upright in a cup, the next day, you're going to bring them in for your display or whatever, and now they look like crap because the stem lying down on the table overnight bent to try to get that cap to, again, be horizontal with the surface of the earth. So it's kind of a neat strategy. Even after they're picked, they still are fighting for the chance to blast their spores and get their genes into the next generation. And polypores can do the same thing too. In the bottom right-hand photo, you've probably seen this. Maybe you knew what was going on, maybe you didn't. Lying down in the forest, you approach a log and there's a big Ganoderma or some other polypore conch hanging off the side. And then weirdly, in a different orientation, there's other smaller conchs hanging off the side of that one. What could have happened? Well, the polypore was coming out the side of the log when it was standing, and it fell over, and now the next year it was still alive. So the next year it produced conchs off of the first one, but in a different orientation, so that again it could keep that cap horizontal with the surface of the earth. Okay, so we're nice and horizontal. It's ready for blast off. Okay, well, what happens next? Okay, so if we zoom into what these Bastidia look like on the surface of the gill, or those tubes of the polypores or boletes, this is what you'd find. Inside that structure, that's where the sex actually happens. The mycelia of two different mating types meet up in the soil or whatever the substrate is, fuse. Sex still hasn't happened. It may never happen, or not until sexual reproduction is going to happen. It can be quite a long time later. It can be years later, and again, it may never happen. So when sexual reproduction is going to happen, that's when a mushroom is formed, and then inside the Bastidia, that's where the sex happens. It's, I'm afraid, not all that exciting, but that's as exciting as it gets. But then comes the blasting of the spores. Okay, so the two different nuclei from the two different parents get together inside that Bastidia, and the chromosomes do the crossing over thing, and you get the genetic diversity, and then they separate after meiosis into the 4 spores, and they get squirted out through that tip there at the aereo called a sterigma, and that's where the spore gets formed. And so now here's when the real magic happens. How does the spore get blasted from the surface? This is how. So if, again, that little horn there, it looks like a crown at the tip, that's what's going to be blown up in the next picture. So the spore gets squirted through that sterigma, and you see this solid red dot in the center of the spore. That's where the center of mass is for the spore. So far, nothing can really happen. I suspect you could probably flick it off there with your finger if you wanted, but it's not going to be blasted off because it's nice and stable. What happens next is on the left side of the spore, this thing called the hilar appendix, there is a drop of liquid that starts coming out in step 2 there, called Buller's drop. Well, while this is going on, there's a sort of a sugary mannitol type matrix chemical that's squirted out of the sterigma there that the spore is attached to, and it goes over the surface of the spore, and this sugary substance is hygroscopic, and thus soaks up water from the air. And more about that in just a second. So as the humidity in the air is sort of condenses on this sugary substance on the spore, and the buller's drop is getting bigger and bigger, you see the center of mass now starts shifting down to where the buller's drop is, because it's getting quite large in step 3. So the center of mass now is way over on the side, but there's some mass starting to happen up near the top because of this adaxial drop forming from moisture condensing in that. Well, at a certain point, the two zones of liquid fuse, the buller's drop coalesces quite quickly with this adaxial drop, and that changes where the center of mass is from down near the bottom left to up near the top of the spore. Quite, it's almost instantaneously, and that sort of catapults in step 5 the spore with tremendous force off the basidia of the spore. And it flies extremely fast, but a very short distance until drag takes over, about halfway between the gill it started on and the next gill, and then it falls down between the gills and walks away. So it's pretty amazing. Buller's drop is named for this famous mycologist of the same name. And he hypothesized a long time ago that this drop forming must have something to do with this, but of course he couldn't see it happening because there's no way to see it happen. It happens so quickly and didn't have sophisticated enough microscopes or anything at the time. But turns out everything he hypothesized a long, long time ago turns out to be exactly true when scientists have looked into this from all sorts of different angles. I mentioned this material, this mannitol that's squirted out onto the cell that's quite hygroscopic and sucks up water from the air. Believe it or not, scientists have actually taken spores off of the basidia and washed them to remove whatever was on them and found out that's what was on there. They've also hypothesized, well, maybe this material is coming from the spore itself. No, maybe it's coming from the sterigma on the basidia. Well, through close examination, they've determined, no, the material is not coming from the spore itself. The spore never changes in size. We know where this material is coming from. So they've pretty much teased out all these different steps over time, but it was quite the genius of A.H.R. Buller, the original mycologist, that kind of saw this and figured this must be something going on. So despite knowing everything about this, we still don't know everything about this. There's Ballistosporic basidiomycetes that they can't possibly use this method to get rid of spores. Here's one right here from the Pacific Northwest. This fungus is Satharella aquatica. As amazing as this looks, this is not a gimmick. This fungus really does rot wood like other Satharellas, but it does so underwater. It's ballistosporic. It has the gills and the spores and the whole mechanism, but no one knows how it works. In fact, the whole reason why basidiomycetes that do ballistosporic, jettisoning of their spores, the why the mushroom cap looks the way it does is actually, for a reason, and it's to keep water off of the hymenium where the spores are being formed, because if rain or a lot of water got on there, of course, it's going to diffuse this whole Buller's drop thing, and it's not going to work. Furthermore, it kind of helps to keep it kind of more humid there so that this process can happen. And another thing is a lot of times spore release by basidiomycetes is at nighttime when humidity is higher, et cetera, et cetera. So all this has to happen for this to work. It can't work underwater, so no one knows how this fungus can possibly release spores. Rafts of spores have been seen in water near these fruit bodies, so maybe air bubbles or something has something to do with it, but there's still lots of other questions. When the spores, if they're released into air bubbles in these rafts that have been hypothesized, well, then what next? Don't they flow on downstream? How does this thing stay in the stream? How does it move upstream? So Jonathan Frank, the guy that discovered this in Oregon quite a few years ago, and it's been seen every year since then, he's noticed a number of different arthropods that seem to be attracted to these mushrooms when they're fruiting. And that's how I first heard about this, because he contacted me, because I've long studied arthropods that have associations with mushrooms. So he contacted me and was asking, does this thing eat mushrooms? Does this eat mushrooms? And turns out all of the different arthropods that he showed me, there are species, of their kind that do eat mushrooms. So he's thought maybe how this thing now has to get around, since it has this aquatic lifestyle, is through a lot of immature insects that live in the water are eating the mushrooms and moving the spores around that way. So jury's still out. Nobody knows. Gastromycete fungi like puffballs is a good example. They don't do this either. So, puffballs, truffles, things like this have evolved. Other sequestrate fungi have evolved mechanisms to allow them to probably move into more dry parts of the planet. And well, they can't really do the Ballistosporic thing because, of the lack of humidity and stuff like that. So they often just kind of keep their spores closed up in an tight, an enclosed chamber, like a puffball or something like that. Truffles, they have come up with a lot of other even more elaborate ways to get their spores around. So, you keep your spores contained in this chamber and even stay underground, don't even fruit above ground. Well, that's great. That allows you to move into drier habitats, and dry habitats all over the world have truffles. Australia is a great example. The vast majority of truffle species live in Australia, and Australia is a very, very dry biome. So, but now that begs the question, well, how do you get your spores around? So they've often had to evolve elaborate ways to entice mammals to dig them up. And a lot of truffles are moved around by mammals. But what if you move to a place where there really aren't a lot of mammals? So like in New Zealand, this is a photo from Noah Siegel. He's studied a lot of different sequestrate fungi, or pouch fungi, as he calls them, that live and fruit in New Zealand. In this picture are all kinds of different sequestrate mushrooms of psilocybe, cortinaria, stropharia. Lots of different species live there, and a lot of them form these sort of pouch-like mushrooms that don't really ever open. And some have stems, some have only a rudimentary little stem, some have no stems at all. So they're evolving sort of a truffle-like lifestyle. And something else you notice about them is, unlike a lot of gastromycetes and truffles and things like that in North America where we live, a lot of these are brightly colored blues and reds. They're pretty cool. And so what could be going on there? Well, what's going on there is, again, they don't have a lot of the mammals that would eat these things and move the spores around, but they have a lot of birds. Flightless birds especially have dominated these niches that mammals fill in other parts of the world. So it's believed that a lot of these fungi, like this Poricotylus pilla, get around by birds picking them up, thinking that they're berries. And this is a really good example here, because in this photo, there's several fruit bodies of the fungus and several fruits from a tree. The fungus fruits at the same time that this particular tree is dropping berries on the ground, and they're basically a dead ringer. So some of them even have sort of a pleasant, almost sweet taste, much like a tree fruit does. So these are sort of truffle-like fungi, but they're not really buried, and a lot of truffles, aren't really underground. Some truffles are on the surface of the soil. Okay, so enough about that. How am I doing on time? Okay.
Fine, don't worry.
Don't worry about it. Well, I have to worry because it's getting late here. It's going to be my bedtime soon. It's not still early in the evening out like it is out where you are. So if you know anything about me, and if the people that have dialed in have seen any presentations I've ever done before, I always manage to work symbionts into the story because I'm just fascinated by them. For 3.5 decades, I've studied insects that have symbioses with mushrooms, and these aren't typically the things that a lot of people know a lot about, and that are frankly a lot more exciting than what I study. I don't look at stuff like termites and ants and things like that are really fascinating. I mostly look at flies and things like that are a lot less exciting, but still they have neat stuff about them. In any case, when it comes to symbioses, fungi are the champs. So as I mentioned, turns out not all fungi, in fact, not most fungi are saprobic. The vast majority of fungi are biotrophic. Another word for that is a symbiont. You hear people use the word symbiont a lot to mean like mutualistically, beneficial. And that's not really what a symbiont is. Symbiont just means two organisms that live in close association with one another. Predator and prey species are symbionts. That's just the way it is. So the vast majority of fungi are biotrophic, and in fact, the vast majority are probably parasites, believe it or not. And if there was a live audience here, I'd probably see a lot of people shaking their heads right now. But the truth of the matter is, the vast majority of life on the planet is probably a parasite. And you can actually just sort of do some simple math to prove this to yourself. Of all the, I don't know, there's like over 1 million named species on the planet, and I think 100,000 of them are fungi. All the 1 million or 1.5 million, I guess maybe it is now, named species on the planet, for sure, all of those organisms have a species-specific parasite that attacks only that species. So think of plants as a really good example. Every species of plant for sure has at least one species-specific parasite that attacks no other organism. So if this is the case, then that means half of all the life on the planet is a parasite. And that's actually kind of, overly simplistic. For sure, all organisms have more than one species-specific parasite that attacks just that particular species. So you can quickly prove to yourself the vast majority of life on the planet is a parasite. And there you go. So fungi shouldn't be any different. But another neat thing about fungi is that a lot are mutualists. Just like plants, their biggest enemy, most of the diseases of plants are caused by fungi. Most of the diseases of animals seem to be caused by bacteria. Most of the diseases of plants are caused by fungi, but at the same time, most of the mutualist partners of plants are fungi and benefit them. So you take the good with the bad. So the neat thing is that, no organism is safe from parasitic and pathogenic fungi. No organism. There's bacteria, slime molds. Here's an example of a slime mold that are attacked by fungi. I don't know if anyone's ever seen this before. This is the common dog vomit Physarum slime mold, which is usually kind of a gray or peach colored or maybe a yellow colored slime mold that oozes along, which is slime molds now, I guess, are protozoans. They're amoebae. Anyways, there's a fungus that makes its living by doing nothing but attacking this poor, lowly, unwitting slime mold, Nectriopus violacea. It's this beautiful purple color. It kind of fades to a light gray. So if you see it, you might have walked right by it and thought, it was just a slime mold having a bad day or over the hill, but it very well could have been parasitized by this fungus that, again, does nothing more than live off of slime molds. Pretty cool. There's, of course, all kinds of different fungi that make their living by attacking other fungi. And so there's some species of this genus, Sporophagomyces, the spore-eating fungi. They make their living by growing as mycelia, as a hyphae on the underside of polypores, and do nothing but just sit there and collect spores dropping from polypores, and that's all they consume, believe it or not. You've probably seen this as well. If polypore conchs are brought into the table during a foray, a lot of times if they have a mold growing on them, they might be old and rotten and might just get tossed. A lot of times they're actually still intact and have kind of a mold on it. They might get tossed because someone thought they were old, but what was going on was this Sporophagomyces growing across the bottom of this thing, harvesting spores. Pretty cool to see. So these are the type of things that are featured in the book. A lot of really neat fungi, and there's of course more to the story on all these, but these are the types of things that often don't end up in books that seem to want to focus on just death caps and stuff like that. Death caps and stuff like that are in the book too, by the way, so if that's what you like. So Puccinia is a group of rusts. Rusts are maybe the most fascinating of all life on the planet. They're these pathogenic fungi, a huge group of basidiomycete fungi that have 2 different hosts in their life cycle, sometimes 5 different types of spores made. Just amazing. And lots of them are economically important. Lots of them, even some of the ones that are economically important, we still don't even know what the alternate host is. It's just so amazing, the variety of rusts. Well, some of the rusts, how they reproduce is by taking over the plant host and causing it to grow these large structures that look like flowers. They're called pseudoflowers. The top picture there in the center, those are inflorescences on the left, but on the right are just these clusters or rosettes of leaves that somewhat resemble a flower. I mean, if you're a bee or some other pollinator, I mean, you don't see things the way humans see things. They see things by bright colors, etc. They don't really see the form so much. So they're very successful at tricking pollinators into pollinating them. In fact, some of the species that attack some of these sort of high elevation plants in the Rocky Mountains produce rosettes that are a different color than the actual inflorescence, but they're even more enticing to pollinators than the actual true flowers are, believe it or not. So in these two photos, what's going on here? The two photos show close-ups of these pseudoflowers. You see these pustules because along with looking quite a bit like the flower itself, along with giving off ultraviolet light in the same color that attracts the bees, they also produce these sweet sticky exudates, just like nectar, as a reward for the pollinators. And you guessed it, inside the sweet sticky exudates are lots and lots of spores. The unwitting insect then carries it to the next plant and infects the plant by pollinating the flower that way. There's lots and lots of species of Puccinias that make these pseudoflowers too. There's pseudoflowers that form on ericaceous plants, all kinds of plants. It's pretty neat to see some of these. Some of these are really dead ringers for flowers too. And then, of course, there's all kinds of fungi that have various symbioses, sometimes beneficial, sometimes to the detriment of their arthropod host. Who hasn't heard of Cordyceps sinensis, this famous Eastern medicine? And what's going on here is it's this fungus that attacks this poor caterpillar. And when the caterpillar at the end of the season burrows into the ground to pupate, well, next season it's not going to come out as a moth. It's going to come out as being dead. And this large fungal fruit body is going to come off the side of it, off the top of the head. But there's more to the story than that. People have long wondered, these are worth so much money. They're worth far more by weight than gold. So it leads people in Tibet and Nepal, where these very high elevation prairies are. It's very, very dry, so there's no trees. They're covered with grass, much like the Great Plains in North America. There's all this grass, and this caterpillar feeds on the grass, this ghost moth caterpillar, and then the fungus infects it. And if the people looking for this thing are lucky in the springtime when this fungus is emerging, they can dig these things up and collect them, and they dry them down, and they're sold for tremendous amounts of money as Eastern medicines. Well, it's like the Wild West out there because it's worth so much money that everybody's picking every single one that they can find. And there's real worry that this fungus is going to go extinct because, it's not going to be able to find any hosts. Everyone's going to be gathering up all the caterpillars that have been infected. But it turns out, I don't know about the fate of the caterpillar, but it turns out the fungus is not going to go extinct. And the reason we know this is not going to go extinct is because the fungus doesn't just live in the caterpillar. Turns out the fungus infects the caterpillar because it's already in the grass. Isn't that amazing? This was just discovered recently. So it's always been wondered, this long structure that emerges through the soil and blows spores all around. Well, if there's fewer and fewer caterpillars because of overharvesting, at some point, all the spores produced by one of these fruit bodies, it's not going to find a caterpillar, and eventually there's not going to be more fungus. But searching around in an environment for where this fungus could be hanging out, scientists found out that the fungus actually is inside the grass. So it's amazing to think that the fungus could get into a caterpillar and take over the caterpillar, not kill it initially, even control its mind, to have it dig down into the soil and orient itself a certain way before it pupates and then ultimately dies, and then the fungus comes out. So not only can has the fungus in its evolution figured out how to take over the caterpillar with caterpillar-like chemicals, But the rest of its life, it's living inside the plant and somehow able to pull that off as well. It's just astounding to me. And the more we look, there's more and more stories like this about fungi. It's just amazing to think. Hidden right in plain sight. Well, we all know about this story. There's closer to home. I mean, there's other cordyceps and cordyceps relatives that attack arthropods here in North America, as well as some fungi. Here's what was known as Elaphiocordyceps. Now it's Talipocladium. It attacks Elaphomyces deer truffle down there where that red arrow is. And I don't know if anyone's found this in the Pacific Northwest. They occur across most of North America, not in the Rocky Mountains area so much, but in the Pacific Northwest, and they're actually very common in the East and where I live. So, and how you find these things basically is by seeing the cordyceps sticking up out of the soil. So we have a number of these different types of cordyceps fungi around that attack all kinds of arthropods and even mushrooms, and they're pretty common knowledge. But have you heard about this before? So this is a group of fungi that attack arthropods, sort of like the cordyceps and sort of not. These are in the order of the fungi known as the Labulobiniales, the labules for short. So you've probably never heard of these, and that's too bad, because despite the fact that we have so many different types of fungi that attack arthropods, this, believe it or not, is the largest group of them. There's dozens and dozens of genera and thousands of species of labules that attack all kinds of arthropods. And probably almost nobody listening to this right now, if they're still awake, has ever even heard of them. It's pretty amazing. They were only discovered in the 1800s. Well, where they were discovered in the 1800s, they'd been seen before that, but they were thought to be structures on the insect. They were thought to be appendages, antennae, etc. Most of the time, there's not so many on a single insect as in this picture. This is kind of overkill. Usually there's just a few. Sometimes they're near the posterior end, down near the bottom of the insect, and that's probably a good place to be, because during copulation, another insect of its kind has to come along and rub around, and they probably infect that next guy this way. But if you're wanting to make a discovery of new fungi out there, for anyone that's a newbie and wanting to name a species or something, this is a good group to work with, because lots of new species of this group are found all the time. And in fact, most of the time, how they're found is scientists just go through collections in insect collections in museums, etc. And that's where they're finding more and more of these all the time. They went unnoticed. There was one that was found just a year or two ago in, I think, Scandinavia, on a millipede or something like that. And it was thought to be a different species of millipede, but when the scientist was comparing to millipedes in North America, the person in North America said, "Hey, that structure there you're looking at that's different. That's actually not the organism. That's a labule." So they were communicating via Twitter. Twitter facilitated the discovery. So the specific epithet of the fungus is Twitterensis. I forget what the genus is, but kind of an interesting little nod there to social media, people doing actual science, not just making up stuff like you typically hear, but actually some interesting things go down. Here's another interesting group, again, of an arthropod hidden in plain sight, having an association with a fungus that you've probably never heard of. You have ash species in the West. I don't think that they're involved with this bolete, this ash bolete, Gyrodon marulioides. This is super common in the East, or at least it was up until recently. With the ash trees going away, it might go away as well, because we have a beetle out here killing them. In any case, any lawn, park, in the city streets, in the fall, late summer, early fall, that has ash trees in the East, you would find this big kind of muddy brown bolete fruiting at the base of them. They're super common. And like all boletes, it's mycorrhizal, right? It's a partner with the roots of specific trees. Wherever you have ash trees, you're going to find this. Well, it turns out people have been discovering only recently, not all boletes are mycorrhizal. And this is one that's not. It is associated with the roots of the trees, but it's not actually attached to the roots of the trees. It's attached to a tiny aphid that's on the roots of the trees. It's a parasite on the roots of the trees. Those red arrows there up in the top left show these kind of black gall looking things. if you were to slice that open, you'd find each one of those has an aphid inside of it. The aphid attaches to the roots of the white ash tree, Fraxinus americanus, and it starts sucking away. But before the tree can kick this pathogen out, this fungus, I mean, again, a lot of this is still hypothesized. It's still not even well known, but the fungus seems to latch onto the aphid and encircle it and even entomb it inside of fungal material, forming this gall, which is to the delight of the insect, because now it's protected from other predators and things like that. And as far as is known, this fungus gets all of its nutrients from this tiny aphid. Isn't that bizarre? So again, this has up until now been a super common mushroom, but no one ever knew what was going on with its life cycle. It was just assumed to be mycorrhizal. So getting close to the end of the talk here, since I've talked for about almost an hour. Miss everyone in the SVIMS. This is what things used to be like when I could make it out to the Pacific Northwest. We could move along at one of the myco blitzes or some such. Here we are looking at a bunch of little black earth tongues. Rich would dig out his camera and everyone would be taking photos of the mushrooms or taking photos of Rich and having great times with friends. Since I knew ahead of time, or I strongly suspected that Andy might try to embarrass me, I figured I would have in my arsenal photos of Andy, because while most of the members treat me with lots of love and respect and kindness, this guy has repeatedly tried to kill me. Here was his latest attempt with this. Let me see what the recipe says. Oh yes, his morel and asparagus crispy pizza. Yeah, so the morel turned out to be, I don't know, this guy has a really great book out, but I don't know about his ID skills because I don't think that was a morel. It looks like a big false morel. Anyways, we ate that gyrometer on the pizza. It actually was quite good. No one got sick. I don't necessarily recommend it to everyone because this mushroom is for sure known to be poisonous. That's a big specimen, but we all lived through it and it was quite tasty. Of course, there's always adolphs on the ground somewhere at all times in the Pacific Northwest. Aluna is usually nearby, identifying some very rare and fascinating mushroom that I've never seen before because she knows all of them. So I very much miss having my good times with everyone in the SVIMS club. So other things to do when I'm visiting the SVIMSers is Kevin often takes me around to see great places. I've been to the museum in Victoria several times and love it. But as great as the museum in Victoria is, if you haven't been over to Vancouver to see the BC Anthropology Museum, this is probably my favorite museum in the whole world, and I live minutes from the Field Museum in Chicago, which is spectacular. But this museum is really, really special. I always love going to see it. Inside is this rotunda room with all this art by this just most amazing First Nation artist, Bill Reed. Around the walls are all kinds of metal and gold work. The guy did wood carving and painting and any media, but most spectacular of all is this big, massive carving in the center, the raven in the first minutes. Probably my single favorite piece of art anywhere in the world. So it's one of those things you can't see too many times. Really love it. In any case, when seeing all of the amazing Haida and other First Nation art there, you'll also see this quote on the wall by a tribute to the Haida: "Everything depends on everything else." So this book that these tales have come from tonight opens with this line, and it also ends with this line because it's, to me, probably the most profound statement there is. Everything really does depend on everything else. In the book, there's the closing chapters on the future and fungi and how a number of problems of the world are being solved through fungi, or frankly, just trying to preserve habitat to allow fungi to do their thing. So an issue of Fungi magazine that I published a couple issues back, we had all kinds of articles on threats to the planet and the habitat, threats to our environment, crops, everything, fires, pollution, plastic, all kinds of things. And each article had some things that are being done to remediate some of these problems with fire or plastic, et cetera, using fungi. So it was a really fun issue to do, and it was quite a popular one. So the fire in particular has been such a big deal in North America and even Australia and even Europe and South America just in the past couple of years. As the climate is getting hotter, fires are everywhere. So, you know, of course you can find burn morels. Out here in the East, we don't have burn morels, but we have other morels. So I'm mostly just showing you some, you paid attention so nicely. I'm just showing you some pretty pictures at this point. But the thing with the burn morels is they have a bizarre life cycle in that they seem to be in the soil doing whatever it is they're doing. And then only after fire is when they go into their sexual reproduction mode and then fruit. And of course, when they fruit, it's in profusion coming right from the cinders. And morels aren't the only mushroom that seems to like fire and have it as part of its life cycle. If you've been to burned areas of forest, you'll see other mushrooms carpeting the forest floor too, like these cute little guys here. This photo's from Kathy Cripps. There's other things as well that seemingly only fruit after fire. And they've just been starting to get studied just recently with the uptick in these massive fires. And one, it's just fascinating to know what they do. But another is, you know, can somehow these help to clean up a forest and fix things and return it to normal after the fire has gone through? This is the goal of some of the work. We had some great lectures last year at the Telluride Mushroom Festival on mycologists that work with fire fungi. Here's one that cropped up last year. There were fires everywhere in Australia. In fact, I do mushroom tours all over the world, and we had one big one planned for Australia that we had to postpone because fires just ravaged all around Australia. Well, after the fires finally went out and the rains came back, people were seeing profusion of these massive mushrooms that most people had never seen before. This Lacocephalum mylidi, the stonemaker mushroom. It's called stonemaker because these big mushroom fruit bodies come from these big, giant underground tuber-like structures. And, you know, the Aboriginal people knew about these and would even harvest these big sclerotia and eat them as sort of a kind of a starchy tuberous food. But in modern times with fire suppression, haven't really seen a whole lot of these mushrooms until last year. They were just everywhere. You could see sliced in half. This is what one of these things looks like. So it's pretty cool. And again, it's out in the forest doing its thing, but just not fruiting until the fire comes through. So with the increase of fires, I think we're going to have to turn to fungi and other things in the environment to sort of save the habitat situation from this you know, increased destruction and return things back to the way it was. So everything depends on everything else. Maybe we'll see more scenes like this, which, you know, while it's burned up trees, looks pretty awful, but there's a lot of promise there with this seed bed. And these seedlings were first inoculated before they were planted. They were first inoculated with mycorrhizal fungi, which gives them sort of a head start. So learning about how this stuff works can kind of help remediate these sorts of problems. So I think that's probably the end. So the last slide I'll leave you with is just sort of a pretty slide of BC mushrooms. And cheers and thanks for your attention.
Thanks a lot, Britt. Britt, could you stop sharing your screen down at the right-hand side, bottom?
Down at the right-hand side?
Well, it may be at the top.
Oh, yeah. Yep, that's top.
Great. Excellent. I know it's very late for you. Do you mind taking a couple of questions?
Oh, sure. Yeah, it's not that late. It's 10:30, so it's not too bad.
Yeah, your Central Time. Yeah.
Yeah.
Now, Kurt,
Yes.
you have some questions in your box?
Right now, there's just sort of more comments. I think Kem Luther had a comment about some mushrooms in Banfield.
Kem, do you want to say something on that?
Yes, they're both of the Tolopocladiums that we have around here are quite common in Bamfield. We see them when we go up there in September.
Yeah,
Anybody
cool.
else?
Okay. And
Alan?
Joe Tanny, I think, was making a bit of a humor, I think, about the ladybugs.
No luck. Well, you gotta look pretty close.
Alan?
So if you're seeing ladybugs this time of the year, it's probably those orange ones, and that's an invasive one, and they're the ones that can like to bite your skin. They try to get inside because they don't want to die from cold.
I have a question.
Go ahead.
Do we still use the word conidia for asexual propagules?
Yeah, so conidia is more correct. People will call conidia asexual spores. It's not really a spore. It's a conidia.
Right.
Yeah.
Okay, thanks. That's got it.
Yeah.
Okay, there's a comment from Home to Me. Can you speak to the occurrence of horizontal gene transfer in fungi?
Oh, wait, what was that?
Can you speak to the occurrence of horizontal gene transfer in fungi? This is from Home to Me, whoever that might be. Maybe if you want to clarify that.
So horizontal gene transfer. So yeah, this has become kind of a hot topic ever since, back when I was a student or maybe a grad student, there were, trees of life of how, different things begat other things down through time. And then all of a sudden, a DNA sequence specialist, Ford Doolittle at the University in Halifax, Nova Scotia, looking at a lot of sequences of organisms, started seeing that, wow, a lot of DNA is not necessarily just being moved around vertically from within a species from parent to offspring. A lot of DNA seems to move horizontally from one organism to another. And so a lot of researchers were like, well, okay, that stands to reason. I mean, bacteria through transduction, et cetera, can actually pick up DNA from the environment. That's how bacteria pick up a lot of resistance to antibiotics or genes for breaking down other, novel chemicals in the environment, et cetera. And Doolittle was like, no, I'm talking about, higher organisms seem to pick up DNA horizontally, spread DNA horizontally as well. Unfortunately, within fungi, I don't know too much about it. I'm not sure who does know too much about it. Maybe Shannon, if she can turn her mic on, maybe she has comments. I don't know too much about genes being moved around horizontally within fungi. It's problematic enough trying to culture fungi, especially mycorrhizal types, like Amanitas, which I'm really interested in. They're mycorrhizal, so you can't really cultivate them on Petri plates or whatever. And a lot of fungi can't be cultivated. The fungi that people have targeted for cultivation, for food, or developing beer and stuff like that, a lot of these reproduce asexually. So they're not really in the business so much of picking up genes from anybody anyway. So I can't really comment.
Britt, this is David, about 20% of human genome is viral. And I just attended a Linneal Society talk on viruses, a thought-provoking talk about it dominating the planet, viruses.
Yes.
And you don't hear too much about viruses and fungi.
Yeah, not too much. So it's a topic that's really, virus is a topic that's very interesting to me. I taught microbiology, university microbiology for quite a few years. So viruses are pretty neat. There's some things about viruses. So of course, they're not living, at least not by my definition and by probably most biologists' definition, not living because they're not made of cells. Also, viruses are not, you know, they're not monophyletic. They're not, you know, there's families of viruses and stuff like that, but frankly, it's almost meaningless. Viruses are basically pieces of DNA, sometimes encapsulated in a shell that can be made of one thing or another, and sometimes nothing. Sometimes just a naked piece of nucleic acid can be a virus, and they're infectious, and they can insert themselves into the genomes of their host, and then turn that host cell into, you know, it enslaves it and makes more copies of the virus itself, and that's how they spread themselves. Well, along the way, when they tear their DNA back out of their host DNA, sometimes they take host DNA with it. And then when they pop back into the next host, now they've moved that DNA from that one host cell into a completely different host cell. So that's, you know, that's one way that DNA can be moved around horizontally, as an example. So that's interesting you mentioned that. As far as viruses of fungi, of course, they get them just like all other life on the planet, and other non-living infectious agents like prions, as an example. You know, that's sort of a proteinaceous infectious agent that causes scrapie and bovine spongiform encephalopathy and Creutzfeldt-Jakob disease, etc. It's not a virus. It's not living. It's a type of a protein that causes other proteins to be misshapen, and then it causes the disease. Fungi get those too. In fact, one of the areas where a lot of prions have been researched is by inserting them into yeast and seeing how they work. Because again, fungal cells are so similar to our cells that they're often used as a model to see how stuff works. But there's, you know, as is the case with a lot of pathogens, they're really only studied if they're economically important. So all the viruses that we know a lot about, you know, it's been argued that possibly the majority of the weight of organic matter in the ocean is virus, and much of the matter in the air. There's been new studies showing sucking air into a filter and then sequencing the nucleic acid of whatever the particulates are. The air is full of virus, and not just COVID, but everything.
No.
So there's
Yeah,
lots
true.
and lots of this DNA. It's astounding. But anyways, as is the case with insects and fungi and anything that's a pathogen, really they're only studied if they're economically important. So the few viruses that we know of in fungi are pathogens of mushroom farms, like La France disease and a few others. But there's lots and lots of viruses that fungi get too. And, you know, like, I mean, we're loaded with viruses, as you just said. About 20% of our genome is viral. You know, most of it has no consequence as far as we know, so nobody ever bothers to study it. But it's fascinating as heck to think about, for sure.
If I can get a copy of that Linnean presentation, the Linnean Society is, you know, quite famous for what they do. And it was just the other day. So if I can get a transcript of that, I'll send it on to you.
Yeah, definitely. Thanks.
Very interesting. It was given by a journalist from Delhi.
Oh, interesting. Well, they should know about viruses there.
Yes.
They probably have, the air's probably thick with viruses there. You know, when you have a lot of people living in close quarters, of course, there's, you know, a lot more chance. There's no social distancing in Delhi, I'm sure.
None.
Tougher to do anyway.
I see Kem smiling over there. He always wondered what was wrong with me now, because I'm from Calcutta, and he must be saying that accounts for a lot of this guy.
Hey, so I see a comment here from Gary, a book recommendation, The Tangled Tree, A Radical New History of Life by David Quammen. I very much recommend, I agree with his recommendation there. Tangled Tree is a great one. It talks about a lot of horizontal gene transfer. And yeah, I read that quite a while back. In fact, anything by David Quammen is absolutely great. Song of the Dodo, or geez, what else? I've read so many things by him. Oh yeah, he wrote about he wrote about this doctor in Austria, Semmelweis, and how
yes.
he discovered ulcers. The cause of ulcers was a bacterium. Just astounding. Everything David Quammen writes is great, for sure.
Gary, could you send us that link? I can barely see it, the Tangled Tree, the Radical New History of Life. So if you send that to me, I'll incorporate it.
Yeah, David Quammen, he cranks out a book every few years, but he's quite a prolific writer. So he writes in lots of other magazines, National Geographic and Smithsonian, and he's always very economical with his words too. So he can pack a lot of really good information into a short amount of space. He's one of the best writers.
There's one more question. Is there a succession in fungi populations, such as after a burn fire or after a burn?
Yeah, so good question. Yeah, there's always succession going on in the forest with fungi, plants, everything, at all times. So, if you're a fan of picking wild mushrooms and foraying, you probably know old growth forests really aren't the best place to go for mushrooms. They probably have truffles and things underground. There's for sure fungi there that are mycorrhizal, but the place to go for mushrooms in a forest is sort of like a young to medium age forest. That seems to be the sweet spot for a lot of the mushrooms that we would be after, because stuff just keeps changing over time. And for sure in a forest, it's the same story. So what might be the fungal succession going on after a burn? So when an opening happens in a forest out here in the Midwest or in the Pacific Northwest or anywhere, the early succession plants are going to be a lot of wind-blown plants, unless they're things that could have withstood the fire, but they'll be wind-blown things. Trees that have serotinous cones, so a few conifers have cones that only open after fire, so those will be there too. But a lot of wind-blown seeded trees and things. So out here, for example, after an opening happens, you would have sycamores and elms and ash, trees that have seeds that blow in, maples, like crazy. And okay, so none of those species attract, for the most part, ectomycorrhizal fungi. They attract mycorrhizal fungi, but not things that produce big mushrooms. So they're going to be more arbuscular type fungi. Then over time, the nut trees and things come in, and then that's when you get the ectomycorrhizal fungi that are producing amanitas and things like that. And then, as they start declining, now you're getting a lot of saprobes, rotting wood, etc. So there's always a succession, whether it's from a burn or any other situation. And in fact, there's been a lot, not to keep going on about fires, but it's just been really fascinating. I've been reading a lot lately, just because I didn't know a lot about what goes on with fires and after fires. And actually, a lot of people didn't know what goes on because we've suppressed them for so long. But the thinking now is, instead of trying to suppress fires or this sort of thing, to do sort of these mosaics in the forest whereby you have patches that you allow a burn to happen, or maybe just log it and have openings and then older growth and then younger growth, and sort of this mosaic. That's thought to be the new best way to do this sort of thing, and kind of stands to reason. I mean, before humans came onto the scene and decided, these stretches of forest needed to be managed, and controlled, because these dumb plants don't know what they're doing. We can manage them and they can grow better. Well, they were doing just fine on their own. They'd been doing it for millions of years, and there would be an opening from a fire or a blowdown or something, and those organisms that have been waiting in the soil, seeds been in the soil waiting for decades to sprout and turn into various forest flowers. The mushroom spores have probably been sitting there for decades waiting for the right kind of tree to come along. And, it's just the way it's always been. And in causing these vast monocultures of fairly old conifers. Basically, they're just tree plantations of just a few species. You know, it's like a time bomb waiting to go off, because when a fire comes, and of course it's going to happen, well, now there's no buffers around, there's no other younger trees to kind of act as a bank for the fungi that can then move into these new areas. So the latest thinking is to be try to allow for more of a natural mosaic type approach, which again is just natural succession. It's not really doing anything special.
Britt, I think we let you go to bed now, or go and have another beer.
I was actually going to say, I was probably about ready for another beer because my glass is kind of empty.
I saw it.
Where's Andy's glass? I haven't seen his. Did he turn his camera off? Maybe he went to bed.
Right here, Brett.
Oh,
Empty as well.
Andy, was that a La Fin du Monde from Unibrew?
Oh, it was indeed, Brett, as promised. And yours?
Oh, that's one of my favorites.
And yours?
That's mine was another homebrew Scotch ale, a wee heavy style.
Ah, very nice.
8.3% alcohol. But the Unibrew, that's far and away, in my opinion, and in the opinion of homebrewers, Unibrew is the best brewer in Canada and one of like the top 3 or 4 brewers in North America. Really love their stuff.
All right. Thank you so much, Britt. I'll give you a call tomorrow and follow up. All right.
Okay. Sure thing.
I much appreciate what you've done, and I'm sure everybody is as well. Great. And I hope we see your book around. I know you can order it. I went on Amazon and there is that discount in effect, and it's a very reasonable price. All right.
Yeah.
So
yeah, hopefully I'll see you guys this fall. If
I love you
everyone.
too, Britt. All the
Night.
best.
Brett.
Thank you.
Yeah.
Thank you
Have
so
a good
much.
night, everybody.